a candidate. Okay, thank you. Hereby I open this academic session in which Mr. Andrew Bowman will defend his academic thesis, Identifying the Elephant, Technological Developments Towards the Complete Identification of Lipids. My name is Nelle de Vries. I'm the Vice Dean for the Faculty of Health Medicine and Life Sciences, and I'm chairing this session. So welcome to all. Before we start the actual defense, I would like to invite Professor Bowman to give a short introduction of his work. The floor is yours.
Thank you very much. Welcome everyone to my thesis defense, and let me go ahead and share my screen. So as was kindly introduced, this is Identifying the Elephant, Technological Developments Towards the Complete Identification of Lipids. And so I'm going to run everyone sort of very quickly through the experimental aspects of the thesis in question. And to sort of cover what everything's going to be, we'll start off with high mass resolution. That's going to focus on the 21 Tesla Fourier transform ion cyclotron resonance experiments. We'll move from there into high spatial resolution with MALDI 2 and oversampling. And then we'll move into isomer resolution with ozone-induced dissociation, collision and ozone-induced dissociation, and negative mode ozone-induced dissociation. So to start off with, this 21T MALDI mass spectrometry imaging, we did this in both positive and negative mode. And we're able to separate very highly, very close masses up to a mass resolution of about 830,000 and mass to charge 800, which is right smack dab in the middle of the lipid range. Our mass accuracy on any of this experiments is floating somewhere around 31 parts per billion. And that's over tens of thousands of identifications of 6 different internal standards across 21,000 pixels. Whenever you're dealing with super high resolution data, one of the most important aspects from 21T data is the increased dynamic range and signal noise ratio. So our dynamic range tends to fall around 536 to 1, meaning that we can detect peaks at a count of 1 and counts of 536 in a single pixel. For reference here, our most abundant peak is PC34-1. This is the potassiated adduct. Signal noise ratio of about 2,370 versus the 2-carbon-13 isotope of PE465 plus a proton, which is hiding all the way down sort of in her baseline, but still has enough information to be able to generate a useful mass spectral image. Using 21T data, we were able to find that our average number of peaks per mass to charge unit could go upwards of 19 different peaks in a single mass to charge. And that equates to about 1,800 peaks total in a 300 to 2,000 mass to charge experiment. For this sort of experiment, one of the reasons why we really wanted to focus on it is because of that mass resolving power to be able to separate out very distinct masses that had very nominally similar mass to charge ratios. In this case, this is a 2.4 millidalton split, which is 2 carbons versus sodium and a proton. So on the left, you can see PC36-1 plus sodium, and on the right, you can see PC38-4 plus a hydrogen. And if those are unresolved, you start to lose any sort of real understanding of what you're looking at in these mass spectra. For the 21T, we're able to find about 190 of these 2.4 millidalton differences in the average mass spectrum, and over 1 million times in the entire measurement. For reference, those are this separation down here, and then you can see over here on the right side, this is sort of a more normal split to be able to find in mass spectra and high resolution data. This is 2 carbon-13s versus 2 less double bonds. We also did these experiments in negative mode. For negative mode, the dynamic range is a little bit lower, about 251 instead of 531 to 1. But we're still able to generate useful peaks at the lower end on this. Over here, this is a peak that we're still not entirely sure what the nominal identification is on it. And then up here with our base peak, this is PI38 for sort of one of the most abundant lipids in brain. Looking into this, we're able to separate out these PA46 from a number of other lipids. That's pretty simple in a lot of high resolution experiments, but what's not simple is separating out these two over here on the right, where this is a 1.79 millidalton split, which is basically invisible in anything that's not a 21 Tesla instrument. And if you were unable to resolve those, you can see that you start to smooth out a number of the distinct morphological changes between this plasmalogen and this phosphatidylglycerol. And then the 1.8/1.79 millidalton split is not as common as the 2.4 millidalton split is in positive mode, but there are still about 35 that you'll find in the average mass spectra, which is more than enough to be able to really want to tease apart what you're seeing in those peaks. So this experimental data had high mass accuracy, less than 0.1 ppm for basically all peaks, and down to 30 parts per billion for our internal standards, which allows for high confidence in our database searching and molecular formula assignment. We could get high mass resolving power of greater than 1.6 million at mass to charge 400, and we were able to get very high dynamic range and signal noise. Moving from that into high spatial resolution instead of high mass resolution, we were using MALDI-2 and sort of our brief schematic here. The blue laser is our primary laser beam, which is desorbing analytes from the surface. And then the purple represents the post-ionization laser that passes through parallel to the surface and desorbs more analytes in the cloud that were not ionized. We combine this with our oversampling technique to be able to ionize things that were at the edge of the laser beam that are not desorbed to be able to improve our overall spatial resolution. For MALDI-2, one of the other significant gains is the overall increase in the number of ions you're able to detect. For MALDI versus MALDI-2, the overall signal intensity increases by an order of magnitude. And the number of identifications is not quite as great a jump, but it is still a factor of 4 to 5 times that we're increasing this. And in fact, at 6 microns, the total number of identifications we're able to receive is approximately the same as you would see in 20 microns in normal MALDI. So MALDI-2 gives you 20 to 30 times higher signal. 55 identifications are possible in normal MALDI, and 330 identifications in MALDI-2, including a number of lipid classes that you just do not see in normal MALDI. So for the average brain, you can see that we're able to identify a number of PCs, PEs, ceramides, occasionally phosphatidylglycerols, ceramides, a whole bunch of other things that are just not available in normal MALDI. And this is a technique that is applicable to multiple kinds of tissues, not just brain. You can run it on kidney as well. And you're able to generate very clear images at a 6 micron pixel size from an 11 by 15 micron laser spot, which we show off here by being able to look at a number of the tubuli within a single mouse kidney. Additionally, because of the fact that you open up further lipid classes with the use of MALDI-2, we can image entirely new features. This is coenzyme Q9 and Q10. These do not show up at all in normal MALDI, but they radicalize very readily in MALDI-2. And then you can also see that we're able to separate those out from our vitamin E, which is centered more toward the inner parts of the kidney. One of the other sets of experiments we did in this paper is to look at multiple sclerosis and human brain tissue samples. And we're able to detect a number of oddities with multiple sclerosis that you would not be able to see in normal MALDI. We were able to detect a number of cholesterol esters, which seem to be highly localized to the macrophages that are consuming myelin inside the multiple sclerosis lesion. And point of fact, we're able to detect the macrophages themselves, which are highlighted very nicely here in red in parts C, D, and E. And then those bright green cores that you see there are actually these super dense packets of cholesterol esters that are effectively carbonizing under the MALDI-2 and separating them out. And you can see that here where these cholesterol esters are mostly showing up in the macrophages themselves. And then there are these highly tight bundles here that are these single cholesterol esters. So you can use MALDI-2 to improve your ionization efficiency. Your oversampling deficits are offset by the MALDI-2 gains, and unique features become available thanks to the MALDI-2. Moving right along from that into ozone-induced dissociation. This set of experiments followed up with a set of experiments that were performed by Marty, who unfortunately passed this last year. But in looking at sort of our changes from his data in the OzID in the Orbitrap versus our OzID in the SYNAPT, a 40 by 40 micron measurement took about 12 hours on the on the Elite. And for the SYNAPT, a 50 by 50 micron measurement took about 4.2 hours with a total pixel scan of 0.2 seconds versus his original settings of 10.3 seconds per pixel. Looking at this, you're able to detect monounsaturated lipids, separate out the N7 versus N9 double bond very readily and easily. You can see that in the complete PC34-1 that is highlighted mostly in the outer gray matter of the brain. And then N7 versus N9 actually has a very distinct shift between the cerebrum and the cerebellum. Using this technique, we can also detect polyunsaturated lipids. In this case, I'm showing you PC38-4 and PC38-6. And you're able to detect not just one set of double bonds, but we can see all of the double bonds in those lipids and determine that there are only the omega-6 or omega-3 fatty acids, and that there are no other components other than some monounsaturated contaminants, which are readily distinguishable from the overall omega-6 or omega-3 fatty acid that is our principal component. With using the SYNAPT, we're able to do CID-OzID squared experiments. So traditionally CID-OzID, you can detect stereo numbered positional isomers through an MS3 experiment. With the SYNAPT, because we could only do MS2 experiments, we were unable to do those sorts of experiments. But if we incompletely fragmented the precursor, we could detect both OzID and CID-OzID fragments simultaneously. There is the slight consequence of the non-specificity. There's some low mass fragment ions that are sort of invisible or lost, but we're able to detect both our CID fragments, our OzID fragments, and some CID-OzID fragments all in one experiment, which we can use to recreate what the original lipid looked like. So these dubbed cause-ID experiments, we're able to detect, say, 16 knot versus 16 knot 18-1 specifically of the SN1. We can look at N7 as a component of all the N7 and N9s, and we can also look at the N7 of N7 and N9 that is specifically SN1 18-1, sort of all in a single experiment. And so owing to the dramatically higher density of ozone, we were able to get higher yields of OzID products. This was about 5 to 10-fold higher than compared to those measured in linear ion trap. Pixel acquisition times were as high as 5 pixels per second without compromising any sensitivity, providing a 50-fold improvement in our acquisition rate. And aside from Oz-ID, Cos-ID experiments can be performed, which show further forms to be able to do CID Oz-ID squared fragments, which are specific for the double bond position at the SN1 fatty acid. And then sort of the last set of experiments that we're able to perform and include in this thesis is our negative mode CID Oz-ID negative mode lipids. And this is unique as far as I'm aware in the first set of mass spectrometry imaging data sets to show negative mode Oz-ID. And in this case, we're able to look at a number of different lipids. I'll show you here. This is PE361 in both brain and medulloblastoma bearing brain, where we're able to detect both N7 and N9 peaks. And we were able to sort of tease those out so that you could see that the N7, N9 actually follows a very similar pattern to what you see in the PCs. And then in the brain, the N7 is also localized to the blastoma itself. And you can do this with the polyunsaturated lipids as well. This is PI384, showing both in normal brain, medulloblastoma bearing brain, and kidney. And you're able to detect sort of differences in sort of the overall picture of what you think that lipid looks like with the lower resolution MS1 that you see in this versus say a 21T instrument, but you can detect the specific fragments of the omega-6 for say PI384 and see how that is significantly either different as in the kidney or how it follows a similar if slightly less clear image in normal brain. But that has been a quick overview of everything that went into my thesis. I'd like to thank Ron Herron, Shane Ellis, and Britt Kleiss for a lot of work and effort that went into helping me with this thesis. Some of the collaborators that I had in a number of these experiments, including Jeroen Boogie and Jerome Hendricks over at the University of Hasselt, along with Mansour Hadar. With the Queensland University of Technology, I'd like to thank Stephen Blanksby and Barrett Pode and Mikhail Belov from Spectroglyph LLC. And for the Florida State University for the 21T experiments, mostly Don Smith, although there were a lot of people who helped us get any of those experiments off the ground. And with that, I thank you, and I suppose it's time to move into questions.
And indeed, it is time to move into the questions. And the first question will be asked by Professor Erik Biesen. He's Professor of Experimental Vascular Pathology in our university. Professor Biesen. Please unmute yourself, Professor Biesen, because otherwise we can't hear you.
Thank you very much. Dear candidate, first of all, my congratulations with your thesis. It's really a magnificent piece of work. And it's your work has clearly advanced the mass spec imaging fields to, I would even say, unprecedented resolution and precision. So congratulations with that. And also I'd like to extend my congratulations to the graduation team for this piece of work. So my collaboration with your supervisor, with Professor Heren, I know of at least one elephant in the mass spec room, and this relates to the annotation of lipids. Is this actually the elephant your title refers to, or were you thinking of a whole spectrum of elephants, a whole zoo?
I would say one of the quotes I have listed sort of in the front of the thesis is it's turtles all the way down. So for me, it's elephants all the way down, because there's sort of the idea of being able to identify specific lipids to say differentiate your PC34-1 as a sort of a sum total composition versus the N7, N9 versus the 18-1, 16-9, and then 18-1, N7 versus 18-1, N9. Being able to separate those is one of the major, I think, questions in mass spectrometry. And to some extent, I feel like that question is becoming more and more answered. The second portion of it is being able to determine those specific identities and then to be able to place them sort of within the specific context of where they're located in a tissue. So there's the exact identification of the lipid in question, and then its exact location. And so those two are closely related to me, but they are distinct questions. So there's at least two elephants that we're dealing with here, and then being able to separate those and identify them while maintaining that spatial context is what's going to be like the next step. So in this question, like some of the work that has been done by Stephen Blanksby's group and a few others who are working in the specific identification of lipids, they are further along in sort of the exact identification of lipids than what I was showing here. But most of those experiments are done after extraction. So you've lost all the spatially relevant information. And so being able to combine these most specific spatial resolution with the most specific lipid identification is going to be sort of the next thing that needs to get done.
Okay. Thank you very much for your clear answer, and I fully agree on that. There's still some work in progress because every sort of issue you solve probably exposes a new level of problems. So my question mainly relates actually to this point, the data you described in chapter 3, and I've, through my collaboration with Professor Heeren, I have some experience with how difficult it is to annotate exactly peaks in an lipidome spectrum to specific full structures, even to species that often is not easy, especially in the context of disease, where you often have exotic lipid adducts like oxylipids that make life even more difficult. How do you handle with these exotic lipids in that context?
So in the context of more exotic lipids, I don't know that the experiments I've done are going to be able to truly answer much of that. A lot of what we ended up doing whenever we were trying to specifically identify these lipids, at least at the some composition level, is to be able to look at not just the primary ion, but to be able to look at the isotopes, because whenever you're floating around 830,000 mass to charge resolution in the lipid range, you can tease apart things that are like the sodium versus protonation. You can start to look into whether or not you have like oxygen 18 versus double bond changes. So some of the more precise data in there, whenever you're looking at it, you can start to differentiate between the different kinds of isotopes that you're seeing. And then you can compare those isotopes sort of to each other. The data that we were able to discover in that 21T data set are isotope ratios for, say, clean lipids, where we knew sort of exactly what we were looking for. PC34-1 plus potassium being sort of a prime example because it's so highly abundant. We could see that the ratio of our isotopes is basically exactly what the theoretical isotope ratio should be. So I think
nicely on that was page 57 and 58. That the predicted peaks of these stereoisomers and the isobaric compounds are really close to what you actually measure. My question is then, how consistent is, for instance, the counter-ion distribution over the different pixels? I can imagine that pH, local pH differences, or local differences in sodium to potassium ratio. For instance, intercellular, you have a completely different, almost inverse ratio. That impacts on the consistency of these isomers. And how well can you predict then or infer that this is not an artifactual peak, but really a peak that belongs to this parental lipid?
So to some extent, you're going to have to, like, I make no claims in any of this data essentially to whether or not the ratio between, say, the sodiated version, the potassiated version, and the protonated version are the same, essentially pixel to pixel, because they are going to change. If you are looking in, say, the white matter versus the gray matter, you're clearly going to have different kinds of ratios. And if you're looking in sort of the synovial fluid space, you're going to have very different ratios than you would in the actual tissue itself. So there is an argument to be made that whenever you're looking at any of this data, that you will inevitably have artifacts in the data. I would say that so long as you can determine the identity based on the isotopes that are available, because you have such high resolution data, that you can at least make an informed guess, say per isotope, or not per isotope, per counterion. So you can say all of the protonated ones should have a similar-ish appearance. All of the sodiated ones should have a similar appearance, and all the potassiated ones should have a similar appearance. You will see that because you have such different environments from one type of tissue to the next, that there is going to be a change, but that change is informative in and of itself, even if you cannot say that there is, an exact micromolar concentration of that specific lipid in that location.
So wouldn't you have made your life much easier by not zooming in on lipids, but on peptides or nucleotides, because oligonucleotides, because they are also, from a biological point of view, much easier to interpret? Or are there other challenges that? And this is my final question here.
I would say probably my life would not have been much easier if I had focused on, say, peptides instead, if for no other reason than the introduction of any of the digestions that would be required to be able to look at peptides are going to inevitably distort what you're seeing in the tissue, because you have to inject so much water. You're going to see a little bit more delocalization. And some of the experiments that were in the later chapters, we discussed that, especially with the Oz-ID experiments, where we had to push everything to sodium, and adding, say, the sodium wash as a dip instead of as a spray over the surface completely changes the localization of many of the lipids. So moving to, say, peptides would have a certain advantage in that you would be able to say with much more certainty what you were exactly looking at, and whether or not you had the same sort of microenvironment, but you would introduce a host of other problems.
Okay, thank you very much with this answer. I'm completely contented, and I give the word back to the pro-rector.
Thank you, Professor Biessen, and I sincerely hope life hasn't been too hard on you, Mr. Bowman, while in Maastricht. The next question will be asked by Professor Ellen Blaak. She is a professor of human biology in our university. The floor is yours.
Thank you, Mr. Pro-Rector. Dear candidate, first, I also would like to congratulate you very much with this impressive piece of work with state-of-the-art methodologies for classifying lipids, and also congratulations to your supervising team. I am more into the physiology of lipids and its role in disease, and not that much into the technical details of mass spectrometry. So I learned a lot reading through your thesis. And I also like where you start off with the Indian story, proverb with the 6 blind men and the elephant, and you take the lipidomics as the elephant. But I would like to discuss a little bit with you on the perspective that disease or diabetes is the elephant, and how we're going to unravel underlying disturbances. Because we know, for instance, diabetes is a disease of disturbed lipid turnover. And I would like to discuss with you certain findings and discuss what these methodologies could mean in elucidating the underlying mechanisms.
Okay.
First question is, because you indicated also in one of your propositions that, of course, to extract the sample may be quite difficult. If I would like to look at muscle or adipose tissue metabolism, is that harder in general in preparation for imaging or mass spectrometry imaging as compared to, for instance, kidney and brain, where you refer to in your introduction?
Looking at adipose tissue, I think would be slightly more difficult in terms of whether or not there is mass spectrometry imaging relevant data there. As far as I'm aware, in adipose tissue, there's like white versus brown tissue, but I don't think that there is necessarily that much highly localized information in aggregate. So if you're looking at, say, one particular fat cell, there will be MSI related information in there. But I don't know if there is, if you're looking at, say, all of the fat tissue, say, in the stomach region of a single patient. So for that kind of experiment, I don't know if MSI would necessarily add that much information. In terms of muscle tissue, you would be able to bring in some of that localized information just because you'd be able to look between different kinds of muscle fibers. And in fact, one of the sets of experiments that I did sort of very early in my PhD was looking at different kinds of muscle tissue from athletic people versus people with obese body types. And sort of trying to look for differences between them. And we found some, but not enough to really be able to generate like a full paper on. But there is mass spectrometry imaging relevant data to be found there.
Okay, and just to make that a little bit more concrete, because there's information, but what kind of information is then feasible? For instance, when you would like to have a look specifically at muscle turnover, that may be disturbed in pre-diabetic individuals, muscle lipid turnover. That may be related to the type of lipid. It's in particular decreased when you have, for instance, a diet with saturated fat. And those saturated fats can incorporate then in different bioactive lipid metabolites. And the location of those lipid metabolites is very important. So you also would like to study dynamics. So what kind of design would you advise me, or what's methodology? If I, for instance, to make it more concrete, in particular would like to look at the diacylglycerols and ceramides, the exact composition, incorporation of label, and to investigate if that's disturbed.
So assuming that the location of the muscle tissue is important, that would be sort of a question at the physiological end, I guess, to say whether or not, say, taking muscle tissue from someone's thigh versus from, say, the stomach is important. That's at that level,
of course, I think that's very important. But I mean, just looking at one particular location of muscle tissue, but the location of the lipid metabolites within muscle, within a particular type of.
If you're wanting to look at ceramides and some of the I guess mass spectrometry imaging are the lipids that are less available at MSI, you would be able to do MALDI-2 imaging because we're able to see the ceramides and you're able to get much greater abundance information out of MALDI-2 experiments in that case. So you would be able to do on, say, like the Orbitrap Elite, a set of experiments that would be able to identify a greater number of lipid classes while being able to, in our current setup, get down to about 6 micron. So for muscle cells, that should be a decent resolution, I would think. Although I will note that one of the things that is happening currently in the lab is we're looking to replace the front end on that instrument with one that will be able to go down to at least 1 micron, if not smaller, in which case you'd be able to look at a greater wealth of information in the intracellular regime, essentially. But I would say for an experiment where you're specifically looking for like ceramides, and if you're only interested in perhaps sort of some composition of the ceramides because you're looking for either total number of double bond information or for elongation information, then you wouldn't need, say, OzID to be able to tease that information out. And MALDI-2 would be able to do everything you need, essentially. So you would need to just take your thin tissue sections. Probably for your patients, you would need to take multiple biopsies over time to see how their microenvironment is changing, and then compare that to other health information to be able to say whether or not they're developing, say, diabetes, or if they are sort of returning to a healthier lifestyle. And in that change, you are seeing a lower incidence rate of diabetes for them so that they are becoming healthier.
Yeah, and that all sounds very interesting. And just to proceed also with an exact experiment, when we would label fatty acids with the 13C label, would you then be able to detect that in the different lipids?
A carbon 13 label would be no issue, especially if you're doing this experiment on our Orbitrap Elite, because typically speaking, whenever you've added so many carbon 13 labels, you are dramatically shifting sort of your mass in the mass charge window. So you're able to see those with relative ease, especially if there are any sort of confounding peaks that you might expect to see close to your original peak, say the all carbon 12 version. So if you are labeling a specific fatty acid and you're looking for that specific kind of turnover, then you should be able to very readily detect that with a MALDI-2 experiment on the Orbitrap Elite.
Okay, thank you very much. Interesting to discuss further, but I think I have to give the word back now to the director.
Indeed, thank you, Professor Black. Mr. Bowman, I must, as Master of Ceremonies, remind you that it is customary to greet the opponents before you give your answers. I think the Beadle must have instructed you about this. And this is extremely important because we are now welcoming a guest from abroad, and that's Professor Gavin Reid. He's a professor of bioanalytical chemistry at the University of Melbourne. Professor Reid.
Thank you, Mr. Pro-Rector. Andrew, I'll start by congratulating you on your presentation this evening here in Melbourne at least. I'll also comment, I met you first and had an opportunity to meet you early in your time as a PhD student. So I'll also congratulate you on how much I think you've matured as a student and as a scientist during that time. It was a real pleasure to read your thesis. So congratulations on that. I'd maybe like to start asking a question, more sort of a high level view of the research that you did that relates to the work you described in chapter 4 and 5. So chapter 4 was really focusing on the aspects related to how to improve the spatial distribution and assign the spatial locations of lipids. While chapter 5 focused on being able to clearly define to improve the structural resolution, at least the detail in which we could annotate a specific lipid located. My maybe overly simplistic view is that those two are almost opposed to each other. You've developed the MALDI-2 method and the oversampling method as a way to improve the spatial resolution, but of course then to obtain an image is going to take you longer. Are those two methods currently compatible in terms of being able to do high structural annotation and high spatial resolution in the same experiment? And what technical advances remain to be made in order to meet that need?
Thank you for the question, esteemed opponent. I would say with the current setup that we have at M4I, the set of experiments are not necessarily inherently compatible, although with the ability to, say, swap out the front end that you would find on like a Synapt instrument, the overall increase in speed that's available from the increased ozone pressure in that kind of system means that while you would see longer experimental times because of your higher spatial resolution in multi-tube oversampling, the total increase in per pixel sort of information shouldn't be that extreme, especially because in the case of, say, running one of these experiments on the Orbitrap Elite, we were floating around 1.1 seconds per pixel. And with the Synapt, we were down at 0.2 seconds per pixel. So assuming that you could essentially put a MALDI-2 platform on the front end of a Synapt OSID, you should be able to do at least one kind of lipid in both high spatial resolution and high structural resolution simultaneously. The thing that's going to be a major sticking point, I think, for any of this is going to be that at least in this sort of experimental setup with OSID, you are limited to one kind of lipid per experiment for the most part. You can do some triplicate kinds of scanning. So you could say do 3 different lipids that are 6 micron in one direction, and then do 18 micron in the other direction to sort of get 18 micron square pixels at the end, where all of the information is sort of collapsed into those. But that's sort of the limitation of what you're going to see with this sort of OSID experiment.
So maybe I'll throw another twist into that. It was, I believe, the review article that you published in the Trends in Analytical Chemistry review article. You also introduced efforts that other groups have taken to be able to understand the dynamics of lipid turnover in an imaging context. Where do you see the ability to then, and of course that's absolutely critical to have the spatially resolved information, the structurally resolved information, and the dynamic information in order to be able to retain some physiological or pathological insights. So, so where do you think the field is at, being able to incorporate really that critical third dimension, which is turnover?
That one's a little harder for me to answer. I would say that I would think that at the current point in time, there is not necessarily any major impediment to being able to combine all 3 of these into a single experiment. The increased time that you would see because you're looking at say smaller pixel sizes combined with the slight slowdown that you might see from being able to do OSID, those are essentially, I think, a separate issue from the number of experiments you would need to perform in order to truly understand what your lipid turnover rate is. So at least to sort of my understanding and being able to do that sort of experiment, you would effectively just need to do different time points sequentially. And so if you were looking at doing, say, the complete identification of PC34-1 in the development of Alzheimer's disease, then the major problem you'll run into is simply the number of experiments that have to be run simultaneously and the amount of data that you're going to generate whenever you do that. So I would say, I think the field as a whole is to a point where it should be able to combine all 3 of these. I just think that there is not yet necessarily one group that has all of the necessary like puzzle pieces to be able to do it.
Okay, so so you showed in the start of your presentation the use of the 21T FTICR instrument to resolve, really that isotopic fine structure. What complexity would then, if you wanted to introduce a dynamic aspect to these measurements, if you're incorporating C13 or deuterium, to what extent would that further complicate or increase the resolution requirement for a mass imaging experiment?
So honestly, if you incorporated carbon-13 to be able to look at the turnover rate of a specific subset of lipids, your overall resolution requirements might actually decrease just a little bit because of the fact that you are effectively moving the lipid of interest away from everything else, essentially, because the mass defect of multiple carbon-13s is going to be very different than, say, a single deuterium or two hydrogens and a sodium. So you'd be sort of sliding back toward that 8.9, 12, 15, 20 millidalton range, depending on how many carbon-13s you're adding. So your overall mass resolution requirement actually goes down if you incorporate that, which makes it a little bit easier, because if you were, say, running an experiment that would be a MALDI 2 experiment on a 21T instrument with ozone involved, you can shorten your scan time by effectively incorporating all these carbon-13s to move it away from all the competitors.
Sure. Yeah. So I think that's, I mean, good answer, assuming that it's a uniform incorporation. Of course, that's going to depend on your carbon source. I think deuterium is another issue, again, depending on the source of deuterium you might use, as to whether you essentially smear your signal out across multiple different isotope-containing species. Yeah, I guess that's maybe something to further work. Can I ask another technical question about
very short question then, Professor Reed.
I'm sorry?
A very short question then, please.
Oh, okay, yep, quick question. You showed in the ozonolysis experiments that a cleavage, that pair of product ions you generate from that fragmentation does not result in two products formed with similar abundances. What causes that asymmetry in the product ion abundance?
The asymmetry that you're seeing, are you talking about specifically how the 16
with a pair of N9 pair of fragments? Why are those fragments not the same relative abundance?
They're not the same relative abundance. I think based on sort of the dynamic of how the ozone cleavage happens, there is sort of a greater preponderance for the subsequent loss of that oxygen, whether it's on one side or the other. So while you will see in a number of cases, you'll have both oxygens on one side. If you have both oxygens on the other side and you lose that second oxygen, you never know it. And if you lose that second oxygen on the first side that still has the charge, then you'll see a lower abundance essentially of that that particular fragment. So you should see sort of a general trend toward the single oxygen component, unless you are over-oxidizing everything, in which case you get into a lot of weird secondary chemistry and your ratios start to alter as well.
Okay.
Okay. Thank you.
Thank you, Professor Reed, for your contribution. I now turn to another guest. That's Professor Edwin de Pauw. He is a professor of physical chemistry and mass spectrometry at the University of Liège. The floor is yours.
Yeah, I have two questions, let's say, and maybe a small one. The first one concerns the number of analytes you have when you do such a high result spectra. In any case, the analytes are there, but you have a lot of peaks. And the question is, what is your workflow? What kind of workflow would you choose between single compound analysis, let's say targeted compound analysis, or family targeted analysis for biological questions? Is it really useful to have single compound analysis, or is it better to have families?
Thank you for the question, esteemed opponent. I would say in general, if you can look at an entire family all at once, that would be more information is available, so you should see more specific information that you can pull out because you have more components to look at. That is not to say that having single component analysis is inherently flawed in and of itself. While you can, while being able to look at, say, all of the 18-1 containing phospholipids is more informative than looking at a single 18-1 phospholipid species. In sort of some of the experiments we've done, especially now that we were able to look in negative Oz ID mode, the 18-1 containing species all show a similar trend. So being able to look at one component tells us more about the whole even if, say, you would have to do multiple experiments to be able to prove that later on, assuming that this kind of trend follows through, even just doing the one experiment is more useful than having no experiments whatsoever.
Okay, so I was just thinking about using a little bit more the Kendrick-Mass defects, because that's a way to fast and to have a fast analysis of your data in such cases. But it's okay. The second question is, what's the real advantage of the 21 Tesla magnet? We are for the moment making a paper on the comparison between different magnet size, and really you got a below 1 million resolution if I look on your data. So what is the real gain? Because, okay, there will never be a lot of such instruments. And if you look the new harmonic detection system, you get the same resolution. And if the cell is becoming bigger, you will have the same signal to noise. So what's the real advantage of the 21?
The real advantage of what you're going to see with the 21T versus a smaller magnet is going to be that your mass accuracy is going to be more stable over long periods of time. And whenever you're dealing with mass spectrometry imaging experiments, the instrument being stable for very long periods of time is one of the most important factors, especially whenever you're looking at this sort of super high mass resolution information. So going to a higher magnet size should be able to better improve your overall experiment by essentially ensuring that your conditions at the start are the same as what they are at the end. I know all magnets have a little bit of mass drift essentially over time, and higher Tesla magnets have less drift than lower strength magnets. That's sort of...
Okay, let's agree with that, but then why don't you use a recalibration?
Could you repeat the question?
It's not a matter of seconds, it's a matter of days. So what is your calibration strategy? Can you not correct that by simply having an internal calibration, not based on standards, but on internal standard, let's say native compounds, just to correct that without the need of a 21 magnet?
I mean, yes, you could add internal calibrants, or you could you could use something that is in the experiment as an internal calibrant to help correct for that. In which case, overall, the only other thing you're going to get out of having a more powerful magnet is that you're going to have sort of a higher dynamic range, and you're going to be able to do the experiment with theoretically the same level of mass resolution faster. But for
when I saw the data, there is only 4-point sampling in the peaks. In mass spec, the peaks 4 points for sampling for one peak. It's a little bit low. And if you increase that, you increase exponentially the time. So, okay, but that's something to discuss. That's a really short question. What about radical ions in MALDI 2?
I mean, you generate a significant number of radical ions, and they're the vitamin E versus like coenzyme Q9 and Q10 are all radical ions. So you do start to see those, and it's actually sort of one of the problems, essentially, at least from the set of experiments I did in MALDI 2, is because if you are looking to compare that to most databases don't include the radical ion as an option to see. So we were in contact and should still be in contact currently with the makers of LipoStar MSI to be able to add radical ions as an option to be able to look at those. So it is a current problem, but there is a solution theoretically on its way. Okay, thanks a lot. Really nice thesis. Really appreciate reading and the content. Nice. Thank you.
Yeah. Thank you, Professor De Pauw. I now turn to Professor Honing. He's a professor of systems imaging at our university. Please, your question.
Thank you, Mr. Prorector. Dear candidate, first of all, congratulations, like all the others, on this beautiful work. And I really liked it. As mass spectrometrist, there's a lot of science and technologies in it. So that always takes care that my heart starts to bounce more than others. But still, of course, I have quite some questions, and I always try to start with some definitions. And on page 13, you say actually that some of the lipids have chirality. But do a molecule have chirality, or do they have a chiral center? It's just of course, on textual, but what's your opinion? Is a molecule having chirality, or does it have a chiral center?
Thank you for your question, esteemed opponent. I'm afraid I'm going to have to say I'm not entirely certain that I have a fine enough understanding to be able to differentiate the two between saying it has a chiral center and saying it has chirality. I would say in the case of most phospholipids, since there is only one chiral center, as far as I'm aware, or there can only be one chiral center, then they would have chirality sort of by definition. If they had multiple points or if they have multiple chiral centers, I guess you could say that they have chiral centers instead of chirality, but I feel that the distinction essentially in the case that I'm dealing with here in this thesis is effectively moot.
Okay, yeah, but that brings me to another question, and maybe that's to educate myself. There's this SN numbering or stereo numbering, and if it's always the second carbon which is chiral, who cares? But then still you have an example of two different molecules, and you say, well, it's actually one palmito, two oleo, and the other one is one stereo, two oleo. And I could not figure out what the difference was. But then when I look into the numbering of how a phospholipid is for example, annotated, you have typically this R and S annotation for the chirality. So what is the difference? So why should I use this stereo number? What does it tell me?
So the stereo numbering, if you're looking at sort of the like triglycerides or the diglycerides, is perhaps less informative just because of the fact that it won't make too much difference essentially. But whenever you're looking at the phospholipids are essentially always attached from with the phospholipid group at the SN3 position. So being able to distinguish SN1, SN2 in that case is sort of the shorthand to be able to say whether it is the further carbon or the center carbon. As far as I'm aware, in sort of mammalian phospholipid generation, the phosphate group is always SN3, but there are like some forms of bacteria and other sort of far removed species where the phosphate group can be on the SN1 position. And so if you can distinguish that point or that chiral center essentially in those sorts of experiments, then you should also be able to say whether or not there is, say, a bacterial load in whatever you're looking at that is distinct from the mammalian host.
Okay, interesting. Thanks for those discussions on definition. And maybe if I have still 2 minutes to go to page 115 of your thesis, where you show the research you did in the ozonolyse in the eye mobility system of the synapse. And one of my questions is that actually what you are doing, and one of our committee members knows a lot about fragmentation occurring in the TWIMS, when you really pump up the energy over pressure ratio, and that's what you do at very high wave and velocity numbers. You start to induce just fragmentation of the compound. And then when you look to the MS spectra of ozonolyse between and the difference between the elite and the synapse, then your claim is that you gain a 50-fold increase in sensitivity of the ozonolyse project compounds. So my question is, what you would see is a very strange combination of just fragmentation of the sodium adduct, I would say, and also then ozonolyse. So what is first, ozonolyse and then fragmentation, or first fragmentation and then ozonolyse? Because that could be, and how can you distinguish that? And the other question is that if this reaction occurs with a higher incidence, is this a kinetic controlled or a thermodynamic controlled reaction?
So as far as I'm aware, the ozonolysis reaction should happen effectively at room temperature if you exposed carbon double bonds to ozone. So it should be sort of in and of itself thermodynamically controlled, although increasing your overall energy does lead to faster ozonolysis yields. In the case that we're seeing here, we are also introducing energy in fragmentation before those ions ever enter the twin cell. So I would say, generally speaking, what we should be seeing as the product ions out of this is fragmentation first, then ozonolysis, instead of the other way around. You will still see some of the products, I think, the other way around because of the fact that ozone is a large enough gas molecule that there should be some sort of collisional energy that you are experiencing in the twin cell, but it should be the sort of kinetic fragmentation beforehand and then the ozonolysis after.
Thank you.
Yeah, I think that also, completed the answer, which is very good because you heard the beetle cry out that hora est, which means that the time for defending your thesis has now passed. The degree committee will now withdraw and discuss the quality of that defense and also of your thesis. And I would ask you and your company to await our decisions online, of course. So I adjourn. verdict, and taking into account the previous qualifications, the decree committee has decided to grant you the degree of doctor. Professor Heren, your promoter, is authorized to confer upon you this academic distinction in accordance with Dutch university custom. So I invite your supervisor to take the floor.
Thank you, Mr. Prorector. Candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I do.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Andrew Bowman, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, as shown by the beadle. Congratulations, Andrew. I'm going to put off my hat and congratulate you. It's done. Today marks the completion of a little less than 4 years of incredibly productive research in the fast lane. Unlike an elephant that typically move relatively slowly unless provoked, you definitely did not. In under 4 years, you have developed yourself more into a scientific cheetah, I would say, if I stick with the animal analogies. They grow incredibly fast, agile, and can rapidly change direction to secure their target efficiently. Really, everything about them moves quickly, very agile, just as you did in your PhD. Your scientific development over the last 4 years was almost like that motion, silent and making an incredible mileage. The numbers quickly: a thesis, a number of lectures with our collaborators, 14 papers, an impressive number of collaborators in under 4 years, with 1.5 years of a global pandemic. I would say a remarkable and very unique achievement indeed. It all started with when your co-promoter Shane and myself met up with you at the IMSC in Toronto to discuss a collaboration with your, at that point in time, supervisor Wichita, where you're pursuing a Master in Science. That after completing a Bachelor in Arts, something that I now might consider an indicator for creativity, as you have been coming up with creative solutions throughout your PhD. Your PhD started a little more than 4 years ago, on August 15th, 2017. And in those 4 years, you've really developed yourself into an independent researcher, as you've demonstrated again today with a wonderful presentation and a wonderful defense. Whether it were challenges with the Orbitraps, the SYNAP, the OSID, vacuum systems, electronics, sample preparation, you managed to tackle them all, which resulted in the thesis that you successfully defended today. One of the remarkable things of how you do science, Andrew, is that you are a true collaborator. And a testimony of that is perhaps the ratio between the papers that made your thesis and the papers that you published during your PhD in collaboration with others that did not make it into chapters of the thesis. The latter being the higher number. And in a collaborative project like lipids, one of our Interact projects that has funded your research, that to me is a sign of immense success. And we all owe you for making that happen so efficiently. So thank you for that. One particular collaboration that I've personally enjoyed very much, and that was discussed at length today, was the collaboration with the National High Magnetic Field Lab in Tallahassee, Florida, where you spent over a month with our MALDI, or our new MALDI ion source, together with Smith, Shane, and myself. At least I was there for a week or so, using the 21T capabilities to really stretch the boundaries of mass resolution in MSI. I truly enjoyed our time there, personally, but also professionally, because of the fact that you managed to really wield that collaboration into a group of scientists that were really doing something that had never been done before. You were a crucial factor in the success of that scientific mission. It demonstrated your unique experimentation and instrumentation competences and capabilities. As you say in one of your propositions, art is I and science is we. If that applies to anybody, it's you. I would also say it's the strength, not only of M4I, our institute here, but also of modern science, at least in my opinion. Another important element in your project was the connection down under. An intensive collaboration with the group of Steve Blangsby at QUT on ozone-induced dissociation in MSI has really made an enormous impact of the field. Indeed, as you said, you followed in the footsteps of Marty Payne, an Australian friend and M4I colleague we lost to colorectal cancer recently, something that we all still feel very sorry about. You followed in his footsteps, and not unimportantly, what you've managed to do with everything that he generated, you sped up the OzID imaging experiments to a level that is actually now routinely usable in molecular pathology. Your co-promoter, moving to Australia in the last year of your PhD, kept the connection alive. And I'm glad that today we are joined by Gavin Reed, not only for another connection down under, but also Gavin Reed will take over another Maastricht effort. Namely, he will chair the IMSC 2024 in Melbourne, taking over the baton from Maastricht. So Gavin, thanks for being here and good luck with the organization in Melbourne. So the connection will stay active for at least a couple of more years. And you, Andrew, were fundamental to all of these collaborations. In the last years, you've worked very closely on Brits, on lipids, on the orbits, the QTOFs and OzID. And she will continue to develop and apply your technologies in her studies of breast cancer. So I'm guessing you might expect a call every now and then to celebrate your success, albeit remotely, but the continued success. It are all elements of identifying the elephant. I sometimes wonder what mass spectrometrists, and I do now consider you one of those species, have with elephants. John Fenn entitled his Nobel lecture, Electrospray Wings for Molecular Elephants, to describe his invention of electrospray ionization. He concluded that the approval from his peers and colleagues in the Fellowship of Scientists had transformed his wildest dreams into a reality even more unbelievable than flying elephants. I hope that the success today on the identifications of your elephant has also transformed your wildest dreams into reality. Another element of success was the implementation of MALDI-2 on the Orbitrap, an approach that is now copied in a number of places in the world. The demonstration of the increased spatial resolution and increased sensitivity has now enabled routine imaging at the cellular resolution, a new leap in mass spectrometry imaging. You've worked with Brooker on testing the MALDI-2 implementation on the timsTOF Flex at M4I, and that instrument is now in bigger demand than any other instrument here at the Institute, and that's in a larger part you're doing. You've always been engaged with working for the group and enabling research possibilities for the group. And whether it was research, it was also film evenings, getting pizzas, or actively participating in the various group activities. This social engagement is part of your success. You're definitely not a lone cheetah, but more of a pack animal in my mind. And that pack animal spirit has translated in outstanding collaborations, but also educational competences, whether it was tutoring, supervising students, or training new colleagues to use your instrument and your methods. These social skills will take you far in your future career in science. And that career will now take you to AbbVie in the US, where you just moved a couple of weeks ago. And you will start in the next couple of days after we've sent you the signed documents of your thesis. That has already been done, and we've already taken precaution for that, so that you can actually start your this new element in your career. When AbbVie asked me if I knew somebody to work with them on their new Bruker TIMS imaging system, I did not have to think long to point you in that direction. I'm glad to see that it has materialized in your new position as a pharma researcher, where you will undoubtedly continue on your path of success with a change that will enrich the elements of the elephants that you can perceive and sense. I want to end by thanking you for everything that you've realized at M4I and your contribution to the innovations and the success of the Institute. In your farewell get-together, I have already handed you over Prometheus, the M4I medal that depicts Prometheus carrying the torch of knowledge. I hope you will keep on carrying this torch on all paths in science that science will roll out for you. Congratulations once again on your newly obtained honor. And of course, I want to include your Melissa and your extended family in my congratulations. I am sure that they are all very proud of this outstanding achievement that you've reached today. Congratulations once again. And applause from all of us.
Yeah. Dear Dr. Bowman, also on behalf of the Board of Deans and of course the academic community here in Maastricht, I would like to congratulate you on this honor. I must say, I hope you noticed the change in the subtitle of your screen. That's very aptly done by our technicians immediately after you got graduated. So that's, and it's of course very well deserved. I extend these congratulations to Ron here and Shane Ellis and the whole M4I community. And I would like to take this opportunity to again thank all the opponents for their contributions today, and especially, of course, Professor Reed and Professor De Pauw from other universities, our guests today. It's also an honor for you that they have agreed to participate in this session. And by that, I conclude the session. We will see each other in the private room in a few seconds, but again, congratulations, and this academic session is hereby closed.